Siddharthan, mechanotransduction, a paradigm shift in pathogenesis of age-related macular degeneration.
Siddharthan is here, no?
Yeah, Siddharthan is there.
Of a study on the mechanotransduction, a paradigm shift in the pathogenesis of age-related macular degeneration. So what is mechanotransduction? So it is the phenomenon by which mechanical signals are converted into biochemical signals by the cells. So one analogy which helped me to better understand this phenomenon is the microphone. So here mechanical signals or sound waves are converted into electrical signals, where in mechanotransduction cells convert them into biochemical responses. So a dysregulated mechanotransduction has been implicated in several diseases, dysregulated responses to flow in atherosclerosis, to stretch in cardiac hypertrophy, and confluence in cancers. Similarly, the cells also respond to changes in the mechanical properties of the substratum on which they sit. So why is this phenomenon even relevant in AMD? We know that the RPE-Brooks membrane interface is the focal point of several hallmark changes which happen in AMD. So the RPE which lies on the basement membrane will come to lie on these deposits in macular degeneration. So our hypothesis was to study the effect of mechanical changes exerted by these sub-RPE deposits on RPE homeostasis and in AMD. So again, to understand how we went about answering this question, I'd like to come back to the analogy of the microphone. So there's the input, which is the mechanical or the sound waves, and the output. But then there is the crucial role played by the microphone, which has a sensor which senses the changes in the sound waves, and then a transducer which converts them into electrical signals. Similarly, mechanotransduction also has two components: the mechanosensor, which senses the changes in the mechanical stimulus, and the mechanotransducer, which acts on these changes to produce the biochemical responses. So the mechanosensors are multiple, belonging to several families of transmembrane proteins. However, they all converge on two main mechanotransducers called YAP and TAS. So crudely, this is how it looks. There are multiple mechanosensors which react to multiple stimulus. However, they all converge on two main mechanotransducers, YAP and TAS. So irrespective of the stimuli or the sensor. So we thought that this would be a good starting point for our study. So one robust way to answer such complex biological questions is to provide interlocking evidence with in vitro cell culture, animal and human data. So we studied the effect of deleting these two mechanotransducers, YAP and TAS, both in vivo and in vitro systems. So first, in cell culture systems, when we knocked down using siRNAs, we learned that the deletion YAP and TAS reduced RPE viability. So we learned that both these proteins are required for survival of the RPE. So the next is, when we knock down these two, they induced RPE inflammation. The expression of NLRP3, which is a very specific inflammatory agent in AMD, was increased. So this showed that the YAP and TAS knockdown plays a specific role in macular degeneration. So though these findings were encouraging, we wanted to learn what happens actually in a living system. So we knocked down the expression of these two proteins in mice. So W2 are wild type mice, where the expression of YAP and TAS is normal in the RPE, and these are double knockout mice, where YAP and TAS is deleted only in the RPE. So on fundus photography of these mice, we see that the mice where YAP and TAS was deleted, there was RPE degeneration, as evidenced by this local area of hypopigmentation compared to the normal mice. Again, on OCT, the YAP and TAS double knockout mice showed RPE loss and secondary photoreceptor degeneration compared to the normal mice. Again, we then started to study the RPE morphology. So this we used immunofluorescence staining of zonula occludens, which is a cell membrane protein where the RPE morphology is highlighted. So these are normal mice, and these are mice in which the deleted, we can see that the RPE morphology is distorted. And this morphology was similar to the AMD RPE morphology seen in AMD. Interestingly, these mice also showed features of choroidal neovascularization on fluorescent angiography. So from the animal data, we learned that knockdown of YAP and TAS induced both wet and dry AMD phenotypes in mice. This is the first animal model to recapture both AMD phenotypes in mice. So again, encouraged by both cell culture and animal, we actually studied does it matter in humans. So we then measured the expression of these two proteins in the RPE in donor eyes with AMD. As you can see here, YAP and TAS expression was significantly decreased in the RPE in human donor eyes in AMD with AMD compared to normal human donor eyes. So what makes it even more intriguing is that there is this rare human disease called Swainson's chorioretinal atrophy, which is characterized by chronic progressive loss of the RPE. And the recent studies have shown that the mutation in the mechanotransduction pathway actually drives this disease. So this is rare, there are only 100 individuals with this disease. We had the opportunity to examine a donor eye with Swainson's chorioretinal atrophy, and the eye with Swainson's atrophy had similar immunopathological features to the eye with geographic atrophy. So by using a gamut of in vitro and in vivo systems, we find that normal RPE extracellular matrix or basement membrane adhesion signaling is required to maintain homeostasis, and sub-RP deposits disrupt this signaling, leading to RP inflammation, degeneration, and neovascularization. So the past 3 decades of AMD research has been focused on identifying the biochemical components of the drusen, and they've identified a multitude of components, and targeting these individual components has been the dominant therapeutic strategy in AMD over the past 3 decades. But our results suggest that an alternative to this existing biochemical hypothesis, we suggest a mechanochemical hypothesis where, irrespective of what gets deposited, just the mechanical effects of these deposits are sufficient to cause RP degeneration and neovascularization. Thank you.
Presentation was good and path-breaking, but there are many questions.
Yes.
How did you estimate all these things? How did you photograph, and how did you do a fluorescein angiography in the mice?
So actually a spectralis, Heidelberg spectralis is sufficient to do an OCT, sir, in mice.
Okay.
It works very well. Only difference is intravenous injection is difficult, so we give an intraperitoneal injection of fluorescein. So the time is delayed.
And how did you estimate those 2 things which are affected more, YAP and SAP?
So we took for fluorescein angiography, fundus photo, and OCT for both mice, for both groups of mice, for wild type and YAP and TAS. So in human eyes, the way is we collected the RPE, and then we took the protein and the RNA, we isolated.
Where from did you collect the RPE?
In human donor eyes, sir.
Donor eyes. Okay. Right.
So we do OCT of the donor eyes also.
Right. Do you think, like between the chicken and or the hen and the egg, what should be happening first? Whether there is cell dysfunction, accumulation of lipofuscin, and this cell swelling, and therefore this swelling of cell giving rise to these chemical transmitters, or
the source of the sub-RP deposits, there are again 2 schools of thought. Either it's the RP itself, or is it coming from the choroid. Irrespective of where the deposits come from, we think that the deposits themselves are causing the disease. So,
so your hypothesis is that there is a change in the cell
floor on which the cells sit.
Yeah, on the sits, and that therefore these chemical transmitters are released, and that's what they cause the neovascularization or RP atrophy.
Atrophy, both. Yes.
How I understand this is that the signal which causes the deposits, that may itself cause the mechanotransduction issues.
It can be a vicious cycle also.
It can be. It may not be even without those deposits. Have you studied that even without those deposits, you have the same kind of features?
Yes.
We do get, isn't it?
Yes. And now we are trying to study the biomechanical properties of the Brooks membrane to see if there is any difference in.
So that will be interesting. That we see these changes even without those drusens in those eyes that we see so often. Anyways, great.
Thank you.
Good one.